Hello, my name is Dr. Cliff Bell, and I'm a research associate professor in the College of Dentistry. I'm still working from home during the pandemic, but I'm happy to hear from you by email or through the Zoom discussions, and my email address is on that first slide. I hope you and your friends and family are staying safe and healthy during the pandemic. In this lecture, I'm going to tell you about a very important aspect of biochemistry, the regulation of gene expression. This shows the objectives for this lecture. What we're going to do is have an overview of transcription and gene regulation. Then I'll talk about gene regulation in bacteria and separately gene regulation in eukaryotes, because there's some major differences between those two branches of life in terms of the gene regulation. The next slide shows something that you hopefully are familiar with, which is the central dogma of molecular biology. It's really about how information in biology is processed through the sequences of these molecules that have different components. The simple version of it is that DNA makes RNA, which makes protein, which we can see in this slide. In a bit more detail then, first we have the DNA that can copy itself through DNA replication as shown here. Then that DNA can also make a copy of itself as RNA. And that process is RNA synthesis or transcription. Finally, the RNA can read off a code of itself to make the same information is moved into protein as amino acids rather than bases as in the DNA or RNA. So it's not a direct copy, and in fact, every 3 bases codes for an amino acid in the genetic code. And generally then the proteins do most of the work that really needs to get done in biology, like acting as enzymes or being structural proteins, for example, in the cytoskeleton of cells or in the extracellular matrix of the body. That's all mostly done by proteins. So you need proteins to get the real work done, but the information is in the DNA and RNA. And today we're going to focus a lot on transcription because that's where the regulation of gene expression into protein, the final product, is carried out. The regulation happens at the level of transcription. Now, why is gene expression and regulation important, and especially now, to you as dental students? Well, there's development and embryonic development. If you think about it, the embryo starts as a bag of cells that are pretty much identical, but then they go on to differentiate and form all the different organs and tissues that are very different. And all those differentiated cells have exactly the same genome, but if you're looking, say, at odontoblast and a gingival epithelial cell, they're doing very different things. What's controlling that is what genes they're expressing from the genome into proteins. So this process is very, very important. And it's central to this. Finally, secondly, gene expression can change to help cells respond to environmental changes. And we'll see examples of that in terms of bacteria responding to different nutrients in their environment and so forth. And it could also be the case of wound healing where, after a dental procedure, the body needs to heal. And so the cells need to change their genes so they can do the healing process. And in other places in health as a disease, for instance, cancer involves a lot of changes of genes that control cell division or cell migration, and that are dysregulated in some cases. The genes are not regulated properly. Or in immunology, and very important for health and disease, the genes are expressed that control the immune response to foreign antigens. And then there's a very recent example of last year, the Nobel Prize for Medicine went to these 3 workers for working on gene regulation. And that's shown in the next slide, what those workers did. They were studying how cells respond to changes in oxygen in the body. And for example, if oxygen gets low, it might mean that a person is anemic, that they don't have enough red cells to carry the oxygen to the tissues. So the kidney, in that case, increases the expression of a gene called erythropoietin, or EPO for short. And you've probably heard of it as a performance-enhancing drug for cyclists and other endurance athletes. And the Nobel Prize came to these workers for working out the pathway of regulation. There is a regulatory protein called hypoxia-inducible factor 1, or HIF1 alpha. And in normoxia at the top, you can see that this HIF1 alpha is hydroxylated on proline residues by the oxygen. And then it becomes bound by a protein called von Hippel-Lindau protein, or VHL, which then causes it to degrade in an organ called, or a cellular organelle called the proteasome. But in cases of hypoxia, where there's low oxygen, the HIF1 alpha is not degraded, and it translocates into the nucleus along with a protein called ARNT, and causes the expression of genes, and that would include the erythropoietin gene that I mentioned earlier, or, for instance, genes that might cause growth of new blood vessels into a tissue that was lacking oxygen. So this is an important pathway in health and various conditions, and that's how they got the Nobel Prize. Now, we're going to look in more detail at transcription, and it's involved by an enzyme called RNA polymerase. And DNA is, as we've talked about, is double-stranded, but the DNA is actually kind of a strange thing because it's this anti-parallel where the two strands go in opposite direction. And because of the way that the base pairing works, where a base pairs with a different base, the two strands are not, in fact, the exact reverse of each other. There's something called the reverse complement of each other. But they know how to make each other. And in the same way, there's a strand called the template strand that knows how to make the RNA strand. And the RNA strand it's complementary to the template strand, but identical to the coding strand. And so here's the template and coding strand of the DNA, and then you're making RNA that's identical to the coding strand, but it's made by base pairing to this template strand because it base pairs perfectly to that. And what happens in RNA, you have uracil U instead of thymine T. That's substituted every time, but that doesn't change the way it base pairs. Uracil still base pairs to adenine, as does thymine. So it's just, we don't have to worry about that in terms of information. It's just something that is happening in making the RNA. As the DNA polymerase makes the RNA, it unwinds the DNA in a small region so that the bases can be exposed, so that it can know what sequence of RNA it needs to be making. And that's shown in this slide. The transcription bubble is where it's unwinding the DNA. And then as it makes the RNA, it moves along the DNA and copies it into RNA, and the RNA grows longer. And here's a three-dimensional structure of the DNA and RNA polymerase and the nascent RNA. You can see the bubble formed on the DNA. You can see the helical structure of the DNA, and you can see the globular RNA polymerase and the nascent RNA strand coming off as it's being formed, and the RNA polymerase is moving along the DNA. Okay, we've seen that RNA polymerase moves along the DNA as it transcribes. It needs to start at a specific point of the DNA sequence to work correctly, and the place where it binds and initiates transcription is called the promoter. Here's a short definition. A promoter is a DNA sequence that determines the site of transcription initiation for an RNA polymerase. We call the promoter non-coding. It says it's non-coding because it doesn't have a DNA sequence that specifies amino acids in a protein, although that DNA sequence is still essential and it's telling something to the RNA polymerase. The DNA organisms is long and contains many genes, which can be on either strand, as shown in this slide here. In this diagram, we see a section of a DNA with 3 genes. two of them are oriented in the left to right direction on one strand, and the other is oriented right to left, and it is on the other strand. And that's perfectly possible and happens all the time in organisms. The promoters are represented by the triangles in between the two DNA strands. And what we can see is that they're asymmetric. The polymerase binds and then moves off in one direction from each promoter. And RNA is always synthesized in the 5' to 3' direction, which is the same way that DNA is synthesized during DNA replication. So all nucleic acids always made in the 5' to 3' direction. And in fact, then the protein is made along the RNA in the 5' to 3' direction as well. So everything goes 5' to 3'. Here's another picture of RNA polymerase, and we can see a lot of the same things we've seen in some of the other pictures. Here we see a better example of where the 5' and 3' ends of the nucleic acids are. We can see a base pairing of a nascent region of the RNA to the unwound part of the DNA, and this is a template strand that would be base pairing to as it's being made. And we can see ribonucleotide triphosphates are moving into the active site of the RNA polymerase, and that's where they're being added on to the nascent chain as the chain gets elongated. This then shows a detail of the enzyme reaction of addition of a base to the nascent RNA chain. Although in this case, it's actually, the picture shows DNA, but it's basically the same as for RNA, so we can use this. And a nucleotide triphosphate comes along where the base is the specific base to base pair to the template strand of the DNA. So it's one of the 4 bases, but it knows which one is base pairing to the template strand. And then it reacts with the 3' hydroxyl of the growing chain, and it forms a 5-phosphodiester bond with the growing chain. And meanwhile, it loses 2 of its phosphates of the triphosphate that are lost as a pyrophosphate molecule after it's added. So we have these phases of our mRNA synthesis: initiation, elongation, and then termination. And that's starting the RNA, making the RNA chain, and then completing at some point you want to complete making the RNA when you've got the whole protein coding sequence in it. And in the case of eukaryotes, it doesn't actually make mRNA directly from transcription. It's making a pre-mRNA, it's called, most often. But in the case of bacteria, it would make the mRNA directly. Here's again a diagram of an RNA polymerase that's showing various steps in RNA synthesis. Initiation is step 1 to 3, elongation step 4 to 5, termination step 6 to 7. And in this case, you see there's a protein called a sigma factor that is binding to RNA polymerase and helping in initiation. And there's a structure in the RNA called a stem loop structure that is helping in termination. And those are both specific to bacteria as opposed to eukaryotes. So I just want to point those out. I mentioned earlier in the lecture that transcriptional regulation is the most common and general form of gene regulation. And this makes sense for evolution because making an mRNA takes some energy. You have to, you lose some energy, you have to expend energy to make this mRNA. So an organism will in general probably be fitter if it doesn't expend the energy making the RNA, if it doesn't need to make the protein at the time. So, everything's evolving to be as energy efficient as possible for the most part. So, within confines that it has. However, it is true that, there's many other forms of regulation that are known and have been found and exist. and I've listed some of these in the here. They include um, post-transcriptional regulation at the level of RNA processing or stability, translational regulation, where you regulate whether the RNA is used to make protein or not, or post-translational regulation. And in this case, you could think of the example of that we've heard of, the HIF1 alpha, where it's hydroxylated and is degraded in normoxic conditions. So that's a form of post-translational regulation that we've already seen. And then meanwhile, HIF1 alpha is causing transcriptional regulation of erythropoietin in the same general pathway. So both things can happen together. And then I want to mention real quickly, this is something you may run into, if you get into research or you read some research papers. There's now possible, there's many genomes have been sequenced, many human genomes have been sequenced. And that opens up this new way of looking at gene regulation, which is called transcriptomics. So the omics kind of implies that we're looking at everything in the genome at one point, and what scientists can do is measure the RNAs for all genes at once and see which genes are being expressed at the level of RNA, and which ones are going up and down in various conditions. And there's an older way of doing this called microarrays, which was actually a chip that had many molecules bound to the surface of the chip, and you could measure that way. And now a newer way where sequencing has gotten very cheap and very high throughput, and now there's a technique called RNA-seq, where you just sequence all the RNA molecules and count them up to figure out which RNAs are expressed higher and which RNAs expressed lower. And now they've even come up with a way to do the RNA-seq on single cells too, so they can look at populations of cells and how the genes are different in various cells. So these are all techniques that are ongoing and helping to study gene regulation. Moving on, I will focus on gene regulation in bacteria, including general principles of transcription and more specific examples of gene induction and repression. This slide shows RNA polymerase from E. coli bound to a promoter. The two horizontal lines represent the DNA, and we can number the nucleotides in the DNA sequence so that where transcription will start is +1. Then nucleotides to the right that are going to be copied into the RNA or downstream are given positive numbers, and the left or untranscribed or upstream nucleotides are given negative numbers. We see that the RNA polymerase has 4 permanent subunits, as shown in red, 2 alpha subunits, a beta subunit, and a beta prime subunit. Then there's the sigma subunit that I had mentioned briefly earlier. In this case, it's sigma 70. It's shown in orange. And that sigma 70 binds to specific sequences near the -10 and -35 regions of the promoter, as we are shown in this picture. And the sigma 70 subunit is important in initiation. We can look at the sequences of different promoters, and here are a number. We've aligned 13 different promoters they're positioned at the +1 site. You can see is orange there. And then if we look at the -10 and -35 sites, we see some similarities in the sequences. So there's the 13 promoters. There's what's known as a consensus sequence shown in yellow at -10, another consensus sequence shown in yellow at -35, and with 16 to 18 base pairs that are not very similar between the different promoters between them, so they don't matter as much. But those 2 consensus sequences at -10 and -35 are where sigma 70 is binding. So sigma factors act as initiation factors. There are different sigma factors, and the number number of total sigma factors can vary between different species of bacteria. And then each sigma factor is given a number by their molecular weight in kilodaltons. So sigma 70 is 70 kilodaltons in molecular weight. And they can recognize different promoter regions. So it is possible, one form of regulation can be you only make a certain sigma factor when you want to express a set of genes, and you can control that whole set of genes through a sigma factor, and that's used sometimes in gene regulation, and why there are multiple sigma factors in bacteria. To break down initiation more detail, it's been found that what happens is the RNA polymerase, as we see on the top there, first binds non-specifically to the DNA, and it kind of slides around until it finds a promoter region that the sigma factor can bind to and recognize. And that happens, and then you get what's called the closed promoters complex, which is shown in the second part of the slide here. Then there's unwinding of DNA around the initiation site, and that's shown then in the third part of the thing, and by that arrow there, and then that's called the open promoter complex, and then transcription begins at that unwound site. The RNA starts forming, and then you get elongation of the transcript. And as that starts, the sigma factor is released from the RNA polymerase. Then the rest of the RNA polymerase is what does the real making of the RNA, and it moves down the DNA strand and copies it while unwinding a small region along the way to be able to make the copy. Then transcription termination in bacteria, as I mentioned, involves a structure called a stem loop. It is possible to have a specific sequence in DNA that, when made into RNA, one part of the sequence can base pair with another part. And so shown here is an RNA being made off of a DNA. You can see part of it's coming off in the top of the diagram, and then it completely comes off. And when it completely comes off, you see the same strand is base pairing with itself in the RNA. That forms the stem of the loop, and the bases at the end are the loop, connecting the two parts of the stem. And this structure then is recognized by RNA polymerase, which knows to stop transcription and release from the DNA. A phenomenon that can occur in bacteria is transcription translational coupling. In the example shown here, a researcher was able to get RNA, DNA, and proteins all together out of a bacterial cell and look at it in an electron microscope. The DNA is the horizontal line that runs through the entire frame of the picture. And you can see two genes with nascent RNAs coming off of them. And so, by the way, can you tell the direction of transcription for the two genes? And if you said left to right, that's correct. You can see that the messages are very short at the left ends of both genes, and they're getting longer off to the right ends as they're being transcribed along the gene. At the same time, you can see some ribosomes starting to attach at the ends of the messenger RNAs. As I mentioned, the ribosomes also start at the 5' ends of the messenger RNAs, so they're out towards the end, and they're making proteins still as the messenger RNA is getting made. Now, this coupling does not happen in eukaryotes because the DNA is all in the nucleus in a eukaryotic cell, and the ribosomes are in the cytoplasm. So the bacteria have one over on us eukaryotes in this sense that they have a more efficient way to do transcription and translation at the same time. Now I'm going to go into more detail about the basic mechanisms of gene expression in bacteria and these two modes of transcriptional regulation, induction and repression. The trip operon is a classic example of repression. So E. coli faces the following situation. Now, tryptophan is an essential amino acid that any bacteria needs to make its proteins. And E. coli has two ways of getting tryptophan depending on the environment. If tryptophan is present in the medium that the bacteria is in, it can transport it into the cell, or if it's absent, it can do biosynthesis from chemical building blocks. So if tryptophan is present, the E. coli cell doesn't have to do the biosynthesis, and if it's absent, it does need to do the biosynthesis. So this is the decision point. And so E. coli regulates the tryptophan biosynthesis genes, and there's some terminology to describe this. The general scheme is called repression or negative control. And there's a protein in the cytoplasm called a repressor, and it's called transacting because it's not permanently connected to the genes. It's usually in the cytoplasm and can bind transiently to the genes. Well, the DNA sequence that it binds to is called the operator and is called cis-acting because it is permanently connected to the gene. So cis and trans are defining the amount of connection of the element that's operating. And the operator overlaps the promoter region and prevents RNA polymerase from accessing it and shuts off the genes, and we'll see the details of that. This shows the trip operon or tryptophan operon in E. coli. The top shows the DNA, and there are 5 genes shown in red, A through E. Below that is the mRNA in blue. Now, the name operon indicates that there are multiple protein coding genes that are made as a single messenger RNA. And here we can see the messenger RNA contains all the genes A through E. This is normal in bacteria, but is not found in eukaryotes, where protein coding genes are separately transcribed. Below the messenger RNA, we see the 5 proteins being made from that message, and those form the 5 enzymes that they're green, and they form all the enzymes that are necessary to make tryptophan amino acid from the building block precursor chorismate. So the technical name for this kind of gene organization is polycistronic. Where you have one promoter, one mRNA transcript, and multiple genes and multiple proteins. So the protein coding part of a operon is called a cistron, and so polycistronic indicates you have multiple cistrons in a single operon. So finally, we focus in on that the promoter and operator are shown on the DNA up above. As we know, they're not made into the mRNA, but they're there in the DNA, and we're going to focus in on them on the next slide. Remembering the desired outcome from the point of view of E. coli, that if tryptophan is not around, the genes need to be expressed so that we can get the amino acid that's needed. But if tryptophan is around, plus tryptophan, we don't want to waste energy in building blocks making tryptophan that we don't need. So here's a zoomed in look at the promoter and operator region of the trip operon. We see that the promoter goes from -35, as we've seen, to close to the start of transcription, while the operator is in an overlapping region in the middle. It actually goes from about -3 to -20, so that this little diagram is not quite to scale, but it's inside the promoter region. At any rate, there are 2 states. in the absence of tryptophan, the repressor actually is normally inactive and doesn't bind the operator. So RNA polymerase is able to initiate, and as we see, the genes are on there. Now, if tryptophan binds to a site on the receptor, it changes the overall structure by what's called an allosteric effect, where a molecule binding to a protein at one site can affect its structure at another site. In this case, the effect on the other site makes it able to bind to the operator sequence into the DNA. Because that site overlaps the promoter region, it blocks binding and initiation of the RNA polymerase, and the genes are off. Next, I'm going to tell you about the lac operon, which is under both negative and positive transcriptional controls. So lactose is a disaccharide that's made up of one molecule each of galactose and glucose. As the name indicates, it's found in milk. E. coli can use sugar as a source of energy in carbon atoms to build other molecules. Its preferred sugar is glucose, but it can break down lactose into galactose and glucose with the enzyme beta-galactosidase, as is shown here. But breaking down possible scenarios. If glucose is present, it would prefer to do glucose metabolism, so it won't use energy on lactose. However, if glucose is absent but lactose is present, the cell will use lactose, so it will have to make beta-galactosidase and other needed proteins. However, if both glucose and lactose are absent, there isn't any point in making beta-galactosidase, but potentially the cell can use some other carbon source. This is the lac for lactose operon promoter region. In this case, we see the transcriptions start at +1, as we've seen before, and we also see the translation start for the LAC-Z gene at +39. LAC-Z is the gene that encodes beta-galactosidase. We also see the -35 and -10 conserved regions that bind the sigma factor. The lines above the DNA represent so-called footprints, and that which is where proteins can be shown to bind to the DNA. The RNA polymerase footprint in red goes across the -10 and -35 regions, as we know. We also see footprints for two regulatory proteins. The lac repressor binds in a region across the start of transcription, where it carries out the negative control part of the dual regulation. And the cap protein bound to cyclic AMP binds on the upstream side of the RNA polymerase site, where it carries out positive regulation. This is how the positive regulation of the lac operon works. In conditions with low glucose in the cell, an enzyme called adenylyl cyclase is activated. It can convert ATP, which we know is the energy-carrying molecule, and we've also run into as one of the 4 nucleoside triphosphates that are used to make RNA. So that same molecule is also used by this adenylyl cyclase enzyme, and it converts it into a form called cyclic AMP, or CAMP for short, shown there. And the CAMP in E. coli binds to a protein called catabolite activator protein, or CAP. And as we saw for the trip repressor, it causes an allosteric change in the protein. And then the CAP with CAMP bound can bind upstream of the lac promoter, as seen in the bottom. And it turns out this is necessary for efficient binding of RNA polymerase to this particular promoter. And this occurs through protein-protein interactions between the CAP and RNA polymerase. Now to introduce the negative regulation and give a little more nuance than we've had so far. In the first diagram, glucose is present, so cyclic AMP is low. In that case, and lactose is absent. In that case, the cap protein is not bound by cyclic AMP, and it's shown kind of bouncing off of the promoter and not binding to the promoter. And lactose is also absent, and in this case, the lac repressor binds to its operator, which is downstream of the promoter, and so the genes remain repressed. We also see in this case the operon actually has Z, Y, and A genes, but we don't need to really worry about that for our purposes. Now, in the second condition here, glucose is present, but lactose is also present. A couple of things are shown. The more important one is that lactose binds to the repressor. It has an allosteric effect, as we've seen before a few times now, but in this case, it's kind of opposite. Where cyclic AMP binding to cap or tryptophan binding to trip repressor cause DNA binding to occur, lactose binding to lac repressor inhibits the DNA binding. So it's an allosteric change that inhibits DNA binding, kind of opposite to the other two. So in this case, when lactose is bound, it doesn't repress the gene. But because cap isn't bound, doing its job because glucose is present and cyclic AMP is absent, you only get a small amount of RNA made. So the promoter is weak without cap, but not totally inactive, and that's kind of a little additional point, but not terribly important. Finally, in the scenario without glucose, but with lactose. So there's no glucose around, you're ready to use lactose. In that case, you get cyclic AMP because of non-glucose is bound to cap, so that's there and present and binding well. Lactose is also present, and it's binding to the repressor, and the repressor does not bind well. So in that case, you have the positive regulation is on, the negative regulation is off, and you can make a lot of the RNA for the lac genes. And this next diagram is pretty much showing a very similar set of things, except it doesn't show that low level of expression when both glucose and lactose are present that was shown in the last one. Like I said, that's not terribly important, but it also shows the situation that's lacking both glucose and lactose that was not shown in the last slide. That is kind of important. So that's the third level down of those. In that case, the cap can bind, so it could stimulate polymerase, except for the fact that the repressor is binding because there's no lactose. So the gene remains turned off, even though cap can still bind. And that's useful to the bacterium because it won't unnecessarily be making beta-galactosidase or the other lac proteins that it doesn't need because there's no lactose present for it to use. And that is the end of the section on gene expression in bacteria. The next part of the lecture is going to be on gene regulation in eukaryotes. And the topics we'll cover include the differences with bacteria, the chromatin structure. Chromatin is the DNA together with proteins bound to it. We'll look at transcription factors, RNA processing, and DNA methylation. So I'm going to talk about some of the differences between eukaryotes and bacteria in terms of gene expression. I should mention that there's also a third major branch of living things called archaea. They're also single-cell organisms without a nucleus, similar to bacteria. But there's a pretty strong consensus among scientists that evolutionarily they're actually more closely related to the eukaryotes than either eukaryotes or archaea are to bacteria. So there actually are closer to relatives to us than bacteria. And in the sort of ways we're looking at gene expression, they actually, though, they probably look more like bacteria in general. But they're also less well studied, and so that things might not always be clear. There's been a lot more study of both eukaryotes and of bacteria than of archaea, so there's a lot less known about them. Anyway, to move on to this picture here, this is an electron micrograph of a bacterial cell. And this one has two cell membranes, an outer and an inner membrane, which means that it's a gram-negative type bacteria. And the periplasmic space is the space in between the two membranes. It has a cell wall, as all bacteria do. And in this case, you can see that it has a defined structure, the nucleoid, where the DNA is present. But there's no membrane surrounding anything within the bacterial cell in this case. And in particular, the DNA is not separated from the cell by a membrane or envelope. Now, if we look at a eukaryotic cell, we can see the DNA is present in the nucleus, and the nucleus is a defined structure that's separated from the rest of the cell by the nuclear envelope for most of the cell life cycle, except when it's undergoing mitosis. And we can also see various organelles in the cytoplasm, many of which are also surrounded by membranes. For instance, the Golgi apparatus or Golgi vesicles, the endoplasmic reticulum, the lysosome, and the mitochondria, all are membrane-bound organelles that are present in eukaryotes. To summarize then, bacteria and archaea do not have a nucleus surrounded by an envelope, do not have as many defined organelles, especially membrane-bound ones, can do transcription and translation concurrently because the ribosomes and the transcription apparatus are in the same cellular compartment. And they have polycistronic messenger RNAs, as we've seen. Eukaryotes have a nucleus with an envelope, have defined organelles, including many bound by membranes, and often have RNA processing as a required step in gene expression. We'll get to that, and almost always have monocistronic genes. So to look at this thing of poly versus monocistronic, this is a prokaryotic polycistronic unit, and in fact it's the same one we saw before of the trip operon that we saw earlier, and and we also see its mRNA below it. And it goes to make the 5 enzymes that do the biosynthesis of tryptophan. And so in this case, ribosomes actually can come in and bind to 5 different initiation codons within that mRNA at the start of each of the 5 genes, and then make the protein until the termination codon at the end of that 5 gene, and then they come off the mRNA. And so it only takes 1 mRNA to make the 5 proteins. Now, in a eukaryotic cell has a polycistronic, or has a monocistronic genes in general. And there's also a lot of other complications relative to the prokaryotic genes. So here's an example of a eukaryotic gene. In this case, it's beta globin, which makes one of the 2 protein subunits of hemoglobin that's found in red blood cells. The final hemoglobin molecule would have this beta subunit, an alpha subunit, plus 2 heme molecules that were bound to each of the subunits. And the heme molecules then bind the iron that carries oxygen from the lung into the tissues. And that's what this is doing. But this is just the one protein part of that larger thing. And this gene only encodes the one protein. And the sequences that are going to be included in the messenger RNA are actually split on the DNA. And the parts that will go into the mRNA are called exons, as shown here. The gaps that are removed are called introns. They aren't shown there, but would be the gaps in between the exons that don't go into the final messenger RNA. So the RNA is initially transcribed as a longer precursor RNA, and then the introns are spliced out to get to the mRNA. It's called RNA splicing. Splicing can occur in bacteria, but it's rare, more the exception than the rule, while nearly every eukaryotic gene is spliced, especially human genes. And there are sites in the mRNA then, they're in both the precursor RNAs and in the mRNA, called the cap site and the poly A site that are shown there. The cap site is at the 5' end and the poly A site at the 3' end. The way translation works in eukaryotes is that the ribosome binds at the cap site at the 5' end and scans the RNA from the 5' to 3' direction until it finds an appropriate initiation codon. So eukaryotic, and then it makes the protein and comes off. And they never come around to the inside of the message first. So you only have one possible protein that can be made from almost every eukaryotic messenger RNA. Another difference is shown in the control regions that are shown on this figure. So in eukaryotes, in fact, in beta globin, compared to bacteria, they're usually close to the genes, as we've seen in the lac or trip operons. It's tens of base pairs from the start of the gene. But here we have regions that can control the expression of the gene that are up to 50 kb away from the gene, which is 50,000 base pairs. And that's compared to the size of the beta globin gene is actually relatively small. The gene, even with exons, is only about 1,000 base pairs. You can see the 200 base scale marker at the bottom there. And it turns out, actually, although for beta globin the RNA coding part is relatively small, in certain genes in eukaryotes, even the RNA coding part can be tremendously huge. So an extreme example is noted at the bottom, which is the gene for dystrophin, which is a human gene. It's mutated in Duchenne or Becker muscular dystrophy. And for that gene, the precursor RNA is 2.3 megabases. So that's 2.3 million bases for the precursor RNA. And then it's spliced down to a messenger RNA of 13 kilobases. So even the coding parts of the gene can be huge with the introns involved. Next, I will cover chromatin structure. Now, the building block of chromatin is the nucleosome. And the nucleosome is made of DNA that is wrapped around these proteins called histone proteins. And to get a closer view of that, here's a cartoon that shows us zooming into chromatin. First, we see a cell with a nucleus and the chromosomes in the nucleus. In fact, the chromosomes in the form that are shown there are from the stage of mitosis, which is the most condensed that they get. And they wouldn't really be in the nucleus like that, but we'll pass over that. And now we see more closely the structure of a chromosome. Now the ends of the DNA are in this case it's been copied over, so it's two long pieces of DNA that are called chromatids with the associated proteins, and the ends of it, the DNA molecules, are called the telomeres. And the middle, where the two chromatids are attached, is called the centromere. Then you can see there's some layers of coiling of different amounts. And as we uncoil more and more, it starts to have the appearance of beads on a string. And the beads on that string are the nucleosomes. And as we zoom in more and more, you can see the individual histone proteins. There are 8 of them that go up to make the protein part of the nucleosome, and then the DNA is wound around them. And then as you get closer and closer in, you can see the DNA double helix is there, and then finally the base pairs within the DNA. So as we've talked about, chromatin is made up of DNA and proteins. And histones are the major proteins in chromatin. And oftentimes people will lump together there are other proteins that are present, but they lump them together as non-histone proteins. And just to mention a technique here, there's a technique called CHIP, or CHIP, chromatin immunoprecipitation. And that's a modern genetic technique where the protein factors are used to pull down specific regions of chromatin, and then the DNA is sequenced. So you can tell in a live cell where proteins are associated, and transcription factors are turning on their genes, inside a cell. And that's a new technique of genomics that you may run into at some point. Here's another zooming view, a little bit similar to the last one, but showing the sizes of the various structures involved. So first you have the double helix, which is 2 nanometers in diameter, and then that gets wound around the nucleosomes, the sort of bead on a string structure, and the nucleosomes are about 11 nanometers in diameter. Then those nucleosomes get coiled into a structure called the 30 nanometer fiber. That then is organized into loops, which can be around 300 nanometers. In chromosome condensation, those can condense down into the chromatid arms, which are about 700 nanometers across, and then the entire chromosome is 2 chromatids, is about 1400 nanometers, or 1.4 microns across. Now, there are 2 major categories of chromatin, euchromatin and heterochromatin. Euchromatin is a more loosely packed form of chromatin. There tend to be more genes in the euchromatin, and those genes can be active. Meanwhile, heterochromatin is a more tightly packed form of chromatin. It tends to have fewer genes in general, and those genes are often inactive. So an example of this is the structure called a bar body, which is the inactive form of the X chromosome, and that is made into heterochromatin to inactivate it. So in that case, the entire chromosome is made into heterochromatin. Also, there can be highly repetitive sequences that are made into heterochromatin that usually don't contain genes, because genes are normally not highly repetitive, tandem repeat kind of things. Genes have to have their sequence to be able to make protein. And these highly repetitive regions are called satellite DNA. Another place where you find heterochromatin is the centromeres and the telomeres are generally heterochromatin. And if you look in the electron microscope, you can see the heterochromatin as darkly staining material, whereas the euchromatin is lightly staining. And the heterochromatin, much of it is near the nuclear envelope, although it can also be irregularly scattered throughout the nucleus, as you can see in this electron micrograph here. Now, here's a picture of the nucleosome, and a demonstration that of the 8 histone proteins shown in yellow, 8 histone subunits that go to form the nucleosome, and they have tails that extend out from the particle, and the DNA is in the red, wound around the nucleosome core. And then you can see how modification of these tails can then affect their interaction with neighboring nucleosomes and increase the tightness or looseness of the chromatin. And in fact, methylation of lysine 9 on histone H3 is thought to be a key part of heterochromatin formation. Histones can be modified in several different ways. 3 of the major modifications are shown here. Methylation can occur on lysine residues, including that key residue I just mentioned on histone H3. Phosphorylation can occur on serine. And a very important one is acetylation, which, like methylation, occurs on lysine residues and can occur on many different residues in these proteins. And looking more closely at histone acetylation, histone proteins contain a large percentage of the basic amino acids arginine and lysine, which are represented in the diagram here with acetyl groups attached to lysine. Note that diagram uses the one-letter amino acid code where arginine is R and lysine is K, because alanine is represented by A and leucine is represented by L. So they needed other letters to represent arginine and lysine. So basically the basic amino acids are shown in blue in that diagram. And we can see acetyl groups attached to some of the lysines. And one of the reasons that histones bind so well to DNA is that DNA is negatively charged because of the phosphates in the backbone, and these basic amino acids that are common in histones are positively charged. And we see that the tail of one of the histones is modified with acetyl groups in that diagram. Now, if we go down to the bottom of the slide and look at, it zooms in on one particular lysine residue, and there are a few things to notice. One is that the acetyl group changes the positively charged lysine to an uncharged acetyl lysine. So it tends to make the protein less charged as you put acetyl groups less positively charged. Another is that there are enzymes carrying out both addition and removal of the acetyl groups. Addition is done by histone acetyl transferases, or HATs, and removal is done by histone deacetylases, HDACs. As histone acetylation modifies the charge of the histone tails, it creates a looser chromatin structure where genes are more likely to be expressed. So a way that gene expression can be regulated is by activator proteins recruiting HAT, which acetylates histones and makes active chromatin at the top in this diagram, or a repressor can recruit HDAC, which deacetylates histones and makes inactive chromatin, shown at the bottom. Additionally, histone modifications like acetylation seem to cause what are called epigenetic effects. In modern biology, an epigenetic effect refers to something which can be inherited by a cell or organism but does not change the DNA base sequence. As we've seen in the central dogma, the DNA base sequence is really where the information is considered to reside for the most part. So this is kind of a little bit of an exception to that. An example is that if you treat a cell with an inhibitor of histone deacetylase, you might turn on some genes. And those genes actually have been found as they will stay turned on even after the cell has undergone a number of cell divisions and the inhibitor hasn't been around for a long time. Other examples of a similar phenomenon are where genes get inactivated by heterochromatin, and then that can vary a bit from cell to cell, but then the effect gets passed down through multiple cell divisions once it is established. So 2 examples of this effect are, one is a thing called position effect variegation, which is when a gene for, say, eye color in flies gets partially inactivated by a chromosomal rearrangement that puts it into heterochromatin. What's found then is the effect is variable but patchy, because whatever effect happens gets established and then is inherited by the progeny cells. You get patches of different eye color when you put an eye color gene in next to heterochromatin. Another example is with the inactivated X. So we talked earlier about the bar body being heterochromatin. So at some early point in the embryo, one of the two X chromosomes is inactivated, but then that is inherited through multiple cell divisions. So the two X chromosomes each appear inactive in patches separately, which explains how you get calico cats, actually. And it's a coat protein or a coat color gene that is inactivated in the X chromosome. And that's why calico cats are almost always female, because it has to do with X inactivation of their patchy appearance. So the mechanisms for epigenetic inheritance in chromatin and histone modification are not exactly understood at this point. But there's another form of epigenetic modification that affects gene expression that's better understood, and that's DNA methylation. So DNA in mammals is sometimes modified at the 5' position of the base, or the 5 position of the base cytosine, to make 5-methylcytosine. More specifically, only certain cytosines are modified, ones that are part of what's called the CPG sequence. The reason why it's CPG is the P is there to emphasize that it means two adjacent nucleotides on the same strand of the DNA. So they're connected by the phosphate bond, as opposed to a CG base pair, which is also a thing, but involves bases on the opposite strands of the DNA. So it's the same strand versus opposite strand, and the opposite strand ones are not connected by a phosphate. So only Cs that are 5' to Gs along a strand are possibly methylated in mammalian DNA. The CPG sequence is actually rarer in the genome than you expect by chance, except in certain parts of the genome, which are called CPG islands. And here's a map of part of the genome showing methylated and unmethylated the CPG sequences. The methylated are filled in and the unmethylated are empty, and their typical distribution. CPGs and transposable elements, which are DNA sequences that can move from place to place, are frequently highly methylated, and that helps to keep their genes inactive, which can keep them from possibly transposing into essential genes and damaging the organism. Meanwhile, unmethylated CPG islands are often associated with the 5' ends, the promoter regions of active genes, like on the right. There may be other scattered CPG sequences, but often the scattered CPG sequences are often methylated throughout the genome. So here's a quick question that has to do with the mechanism of things. And remember, we always write DNAs 5' to 3', or think of them 5' to 3', the sequence, even though one strand is going one way and the other strand goes the other. So you can consider that question for a couple seconds here. Okay, the answer is that it's always CPG on the other strand too. So it's just going the other direction. But G base pairs with C and C base pairs with G. So CG on one strand is matched up with a CPG on the other strand. Another way of putting it is that CPG is the reverse complement of itself. And this is a handy property for maintaining the patterns of DNA methylation and can lead to epigenetic inheritance. In this slide, you can see a parent DNA here that is methylated at the left-hand CPG, but not at the right-hand one. It divides into two cells, and since DNA replication is semi-conservative, the parental strands in red keep their methyl groups, while the newly synthesized strands in orange have unmethylated cytosines, because that's what's incorporated in the newly made DNA. However, there's an enzyme called DNA methyltransferase in humans, and what it does is it specifically recognizes CpG residues that are only methylated on one strand and methylates the C residue on the other strand, as seen here. So it's only methylating the left-hand one on the orange strand and not the right-hand one on either strand, because that one is not hemimethylated. And this is possible because of the self-reverse complementary property of CpG. A few final facts on DNA methylation and its effect on gene expression. So if gene promoter CpG islands are highly methylated, they're very likely not to be transcribed. But if they are unmethylated, it doesn't guarantee that they are transcribed. So although there are these nice mechanisms to preserve the methylation patterns and inherit them, there sometimes are changes during development in DNA methylation. So it's not something that's totally fixed in stone, but it does change in a programmed way at times. And finally, methylation is frequently deregulated in cancer, so that's an important mechanism in terms of health. And that's what I'll say about DNA methylation. Now I will discuss eukaryotic gene regulation, and I will start with a discussion of RNA polymerase, or Pol2 for short, and transcription factors. Now, this diagram shows various levels that it's possible for eukaryotic gene regulation to occur. In some cases, the steps are similar to bacterial gene regulation, while others are specific to eukaryotes. The gray lines in the middle of this diagram represent the nuclear envelope, with the nucleus to the left and the cytosol to the right. The most common and most efficient method of control is transcriptional, and we talked a little bit about that in terms of bacteria. So how strongly the RNA transcript is made from the DNA. Another level of regulation that can occur is at the level of RNA processing. At this level, you can control not only how much RNA is made, but also whether different RNAs are made from the same gene by variable splicing. Next is the process of transport of RNA out of the nucleus. This can be controlled and can serve as a level of control. Then you can have expression at the level of translation, whether the message is translated into protein. Control of expression at the level of mRNA degradation. So if you change the rate of mRNA degradation, you will change the amount of protein. And finally, the protein can be controlled at the level of its activity. And we saw examples of both the initial step and final step in the story of HIF1 alpha and the Nobel research that I discussed very early on. In the presence of oxygen, HIF1 alpha protein is modified and degraded, so it would be regulated at step number 6 at the protein activity. Whereas then it goes on to in hypoxia, it regulates other genes at the level of step number 1, the transcriptional control. So we can see both of these levels are working in a real situation. Now to go concentrate on step 1 for a moment, that's transcriptional regulation. One aspect of this is differences in RNA polymerases. So as you see here, bacteria have a single RNA polymerase that synthesizes the RNAs for ribosomal RNA, rRNA, transfer RNA, tRNA, or messenger RNA, mRNA. In eukaryotes, RNA polymerase, there are 3 RNA polymerases. RNA polymerase 1 synthesizes ribosomal RNA. RNA polymerase 2 synthesizes precursors that go on to make messenger RNA. and RNA polymerase III makes transfer RNA, and one of the ribosomal RNAs, the 5S ribosomal RNA. And additionally, there are some RNAs that are only found in eukaryotes. The small nuclear RNAs, or snRNA, and microRNAs, or miRNA. They're made by both RNA polymerase II or RNA polymerase III, depending on the specific gene. But since RNA polymerase II is the one that makes the pre-mRNA, that's the one we're going to be concentrating on. This is a fairly busy diagram that introduces the general processes that are going on in transcriptional regulation in eukaryotes. The gene in question here is gene X, and it's going to make an RNA transcript, as we can see there. The large light blue blob is RNA polymerase II, and it's threading the DNA through a groove in the protein, similarly to the bacterial RNA polymerase that we saw earlier in the lecture. Also, like bacteria, there's a promoter that is close to the start point of transcription that the RNA polymerase binds to. It binds at the promoter together with a number of other proteins that are called general transcription factors. And a DNA sequence that is frequently involved in this binding is called the TATA box. You can see that in green there. And it has this, it's called that because it has the base sequence. The TATA is for the base sequence thymidine, adenine, thymidine, adenine. The box is kind of, I think, just a sort of a way to describe a sequence. Okay, finally, there are other regions that can bind proteins and regulate gene expression. and they can be located either upstream or downstream of the transcribed region, and sometimes at rather great distances away from the start of transcription. They bind gene regulatory proteins, and in the diagram are called regulatory sequences. Often they are also called enhancers or silencers, depending on how they affect the transcription levels. And they bind other transcription factors that are not the general transcription factors, or in the diagram they're referred to as gene regulatory proteins. So in the middle part of that previous slide, we saw Pol2 binding to the promoter with some general transcription factors. So we'll take a closer look at that. And there are 5 general transcription factors that are pretty universally used, and others that vary from gene to gene. Now we're going to look at the formation of something called the pre-initiation complex. And in this first slide, you can see at the top the promoter with the TATA sequence. And that sequence is 25 bases to 35 base pairs upstream of the transcription start point. The orange protein, which is called variously TATA binding protein, or TBP, or transcription factor 2D, binds to the TATA sequence. And there are also a number of proteins called TAF, for TBP associated factors, that bind along with the TBP and help it out. And those vary with the specific promoter. Also, some promoters don't have TATA boxes, and TAFs are especially important for those. Next, the protein called TF2B, which is the green protein, binds along with into the complex. At this point, RNA polymerase II makes its entrance together with another factor called TF2F. And then 2 more factors, TF2E and TF2H, are added. And we're ready to start transcribing RNA, starting at position +1, as seen here. And as you can see, a lot of this process is pretty similar to that in bacteria, with TF2D or TPB kind of acting like the sigma factor in the bacterial case, and the Tata sequence being kind of like the -10 and -35 sequences in bacteria. Here's another view that's going to take us through initiation. Here you see the Tata box again at the beginning. Then TBP TF2D binds to it. And in this picture, we see an additional wrinkle, which is that the DNA makes a bend when TF2D binds. This diagram doesn't show the TAFs, though. Then TF2B comes in, then Pol2 and TF2F. And this also shows the C-terminal domain of Pol2 making interactions with the transcription factors. Then TF2E comes in. And following on, TF2H comes in and forms the what's known as the initiation complex. After that, the C-terminal domain of the polymerase is phosphorylated, and the general factors are released from Pol2, and it starts transcribing the gene. And Tata binding protein remains bound to the gene when the other general transcription factors are released. Going back to the overall model for eukaryotic transcription, you can see that we've discussed the central part here with Pol2, general transcription factors, and the Tata box. Now we will turn attention to the outer parts of this figure that actually regulate the gene expression. Most oftenly, these act in a positive manner. The DNA sequences upregulate gene expression and are known as enhancers. Sometimes they can downregulate expression, and then they're called silencers. We'll talk mainly about enhancers because they seem to be more common and are better studied, but silencers are probably similar except for downregulating the gene rather than upregulating it. This shows some of the properties of enhancers. They can often work in different positions or orientation. In part A, there's a gene with its promoter. It does what's called basal transcription. So you see a few transcripts there. In B, an enhancer has been added. The box is labeled E, and it's greatly increased the amount of transcripts. In C, the enhancer is moved very far upstream from the promoter, but it's still increased transcription. And in D, the enhancer is moved downstream from the promoter and far away, but it's still increasing transcription. And then in E, it's far away and it's flipped the opposite orientation, but it still gives a high level of transcription. And people figure this out by doing these kinds of experiments by, rearranging DNA with enhancer sequences relative to the promoter. And it was quite surprising when discovered because the regulatory proteins we talked about in bacteria or the general transcription factors in eukaryotes that we just talked about definitely wouldn't work like this. So if you moved the LAC operator away from the gene, LAC repressor wouldn't be able to control it at all. So what's going on here? One thing that's going on is the DNA is looping around. In this picture, we see Pol2 bound with the general transcription factors at the promoter and a couple of specific transcription factors. Just upstream of the Tata box is a green transcription factor bound on one site, And then further upstream is a turquoise transcription factor bound to an enhancer. We can see that the DNA is looped around to allow the enhancer-bound factor to interact with the polymerase complex. And as mentioned but not shown here, the TATA binding protein bends DNA where it binds, which can contribute to this effect. So that's not shown in this diagram, but we saw that in an earlier diagram. The orange protein is something called the mediator complex that connects transcription factor activation domains to polymerase II. There can be multiple regulatory proteins, positive or negative, bound to various sites, and they can all have DNA loop around to interact with the Pol2 and the promoter and produce the final regulation of level of gene transcription. Insulators can block the action of control elements on genes where they are not intended. This is needed because, as mentioned, enhancers can act over large distances in the genome. In the example here, the insulator is blocking an effect of an enhancer on gene A while allowing the effect on gene B, or separate, and then a separate insulator is also acting to block the spread of heterochromatin, because heterochromatin tends to expand, but insulators can prevent it getting into the gene and activating gene B in that case. Transcriptional activators are modular and have domains, which is true of many proteins, but we're going to discuss it for transcription factors in particular here. And in particular, they frequently have separate DNA binding and activation domains, the positively acting ones. So here we see the DNA binding domain interacting with the DNA in light blue, and the dark blue activation domain interacts with that mediator complex that bridges across to Pol2 and the 5 general transcription factors. And now, if we're looking at the DNA binding domains, there are a few different structural types that are known, and we'll just go through those. So looking at that DNA binding domain, here's a few different structural types. They basically need to be able to recognize specific DNA sequences in order to regulate their genes specifically. So they have to make a contacts within the grooves of the DNA helix, usually the major groove, to sense which bases are present and be able to bind specifically. But with that requirement, there are a number of different structures that can do this, but there's sort of groups of structures that do it. And they're reused again and again. The first one is called the helix turn helix. That's one type. And that includes what are called homeodomain proteins. And some of those act as master switches in development. So when they're mutated, you can have things like a fly with a leg growing where its antenna should be. They control development to that level that they put something in entirely the wrong place. So that's homeodomain proteins. But the bacterial proteins that we talked about in the section under bacterial proteins are all also helix turn helix proteins. So the trip repressor, the LAC-I, and the catabolite activator protein are all helix turn helix as well. Then other DNA binding motifs are zinc finger proteins, the leucine zipper proteins, and helix loop helix proteins. And the HIF1 alpha protein mentioned earlier is a helix loop helix protein. So here's some examples of the the three-dimensional structures of these common domains. So this is one type of zinc finger structure. There's actually a number of different ones. The thing that they all have in common is they all incorporate the metal ion zinc. And in the structures, zinc is coordinated to 4 amino acids, usually either 2 cysteines and 2 histidines or 4 cysteines. And those zinc molecules then fold the protein into a compact domain that allows it to bind DNA. And you can see one example of that here with 3 zinc ions depicted, each one making 4 contacts to the protein, and then it's bound into the DNA. Important zinc finger proteins are the steroid hormone receptors, like the estrogen receptor and the glucocorticoid receptor. Steroid hormone receptors are major drug targets, so they're important in medicine and in dentistry, including for corticosteroids, things like birth control pills, tamoxifen for breast cancer, and on and on. So the steroid hormone receptors are transcription factors, actually. This is an example of the structure of a leucine zipper. It's formed by dimerization of 2 alpha helices that are the blue cylinders. The helices are called amphipathic because they have one side that is hydrophilic and another side that is hydrophobic. And it's hydrophobic because of the presence of leucine residues, particularly possibly other hydrophobic amino acids. And those then form a kind of zipper where they stick between each other, and they stay that way. They stay out of the aqueous solvent, and that's what stabilizes the structure. And then the helices go down and bind on either side of the DNA helix, and it's stabilized through these leucine interactions. Now, going back to this original figure showing the pathway of eukaryotic gene expression and steps of where it might be regulated, I'm going to finish up with a section on the second step depicted here, which is RNA processing. This is an overview of what happens to make a eukaryotic mRNA from a precursor RNA. The RNA is transcribed from the start site to the termination site or sites, possibly. The original RNA is called the primary transcript, as it says there. As we should remember, this occurs in a 5' to 3' direction in terms of the RNA. At the 5' end of the RNA, a structure called the 5' cap is added. Another modification that's made in RNA processing is the cleavage between the translation termination and the transcription termination, and addition of a sequence of repeated adenosine, or A for short, is added to the new 3' end that's created by this cleavage. They're usually between 100 to 250 adenosine residues added, as it shows there. And the resulting structure is called the poly A tail, and the process is called polyadenylation. The third part of processing that we're interested in is introns being spliced out of the transcript. That happens, and then you get finally the mature messenger RNA that has introns are gone. It has the poly A tail, and it has the 5' cap structure. Here's the structure of the 5' cap. It's composed of nucleotides, but it has some unusual features compared to typical RNA. A base is added to that wasn't coded for in the DNA. In fact, it's not even a totally normal base. It's a modified form of guanosine that has a methyl group in the 7 position. So 7-methylguanosine. There's another weird thing. It is connected by phosphate bonds to the transcript 5' end, but instead of connecting the 3' position to the 5' as would normally happen, it connects the 5' position to the 5'. And it also has 3 phosphates in the bond rather than the normal 1 that's found in normal RNA. So it's a 5' to 5' linkage with 3 phosphates. The final part of the modification is that the first 2 nucleotides of the RNA that were made on the DNA template are modified by being post-transcriptionally methylated on the 2' hydroxyl of their ribose. So that's the structure of the 5' cap, and that's something that gets added on, or modified after the transcription starts. Now that we've seen what happens to the 5' end, we can look at the 3' end. I'll just do another quick reminder that we always write and think about nucleic acid sequences 5' to 3'. The nucleic acid gets synthesized in that direction, and the protein gets made in that direction along the RNA. Okay, so in bacteria, we saw that transcription actually terminated at the 3' end of the message, and the RNA polymerase came off at that point. But in eukaryotes, the RNA polymerase keeps going. So RNA polymerase 2 keeps going past the point where the 3' end of the message is and terminates downstream, probably at multiple different points. So the 3' end of the mRNA is generated by cleavage and polyadenylation as shown here. Top shows the DNA and the DNA sequences that are needed, which are an upstream element, a GU-rich downstream element, and the sequence AATAAA, followed a short while later by CA. And that's transcribed into the pre-mRNA with the usual substitution of uracil for thymine that always happens in transcription. Next, it's cleaved by an endonuclease just after the CA sequence. And after that, a string of the nucleotide A are added on by an enzyme called poly-A polymerase, as shown there, which is a kind of RNA polymerase that doesn't copy a template. It just adds the specific nucleotide A up to 200 or 150 to 250. And RNA polymerase 2 terminates at multiple sites downstream after this cleavage has occurred in most cases. The last step of RNA processing that I want to discuss with you is splicing of the pre-mRNA. Nearly all human genes have the sequences that are going to be incorporated into the mRNA interrupted by other sequences called introns. The sequences that are included in the final mRNA are called exons. Now, the top diagram here shows a simplified precursor RNA with 2 exons, one in yellow and exon 2 in green, with the brown intron between them. there are 5' and 3' splice sites, and there are some conserved sequences. GU just after the 5' splice site, and AG just before the 3' splice site. The reaction is carried out by a complex of proteins and RNAs called the spliceosome. The RNAs of the spliceosome are called small nuclear RNAs, or snRNAs for short. And there are two steps in the splicing process. In the first step that we're on now, the bond between exon 1 and the intron is broken, and the 5' end of the intron forms a bond with a 2' hydroxyl of an A residue in the intron, forming a structure that is called a lariat because of its shape with the loop and a stem coming out. And in the next step, exon 1 attaches to exon 2, and the intron is released, still in its lariat form. And so at that point you have, the spliced RNA, and in this case we only had those 2 exons, so that's all we need there. Most cases you would have more introns and they'd all have to undergo this process. Now, one might think that RNA splicing seems quite inefficient. And it's true that bacteria and some eukaryotes, like yeast, don't do that much of it. Probably because it is a bit inefficient to have to do this every time you want to express a gene. But it probably can have some quite useful advantages. I talked in the section about transcription factors about domains and proteins. And one quite handy thing with splicing is that domains can be made either by single exons or groups of exons. And it's then possible to shuffle these domains and mix and match during evolution to get useful functions that are combinations of domains that do different things. And there's also a way to do that within a single gene, sort of in real time rather than during evolution. That's shown here. In this case, it's a gene for transcription factor for a transcription factor with 4 exons. The DNA binding domain is on the green exon, and the activation domain is on the orange exon. Then cell 1 and cell 2 are making factors that cause changes in RNA splicing between the 2 cells. And in cell 1, the gene makes an activator by including the orange exon, while in cell 2, it skips the orange activation exon and makes a repressor that binds to the same DNA sequence because it has the same DNA binding domain. And I'll just mention that in recent years, with the sequencing of the human genome and sequencing of mass sequencing of human RNA, it seems that almost every human gene has multiple differently spliced transcripts. So this is a major thing that is happening in our genome all the time, is alternate splicing of messenger RNA to form different forms of the gene product. So that's the end of the main part of the lecture. I'll go now and recap what I've told you. So we started just talking about the importance of gene regulation in general in biology, and hopefully more specifically to dentistry and medicine. And I gave the example of HIF1 alpha and how that won the Nobel Prize and how it's important in regulating the body's response to levels of oxygen and controlling various things based on that. Then I talked about general aspects of transcription in particular, because transcription's a major part of gene regulation, probably the most major part, and RNA polymerase, what the reaction is, and things like that. Then we talked about bacteria, um, things like promoters, sigma factors, termination of transcription, transcription and translation coupling is possible. I went into regulation and gene regulation in bacteria, how there are operons. The operator is the sequence that things bind to control the expression of the operon. And a couple of specific examples. The trip operon is an example of repression. And then after that, the lac operon involves both induction and repression. And then we talked about switch to eukaryotes to talk about gene regulation, about differences between eukaryotes and bacteria. I talked about chromatin structure and how nucleosomes are the basic building block of chromatin. The histones make up the nucleosomes. You have different forms of chromatin, and the major branches are euchromatin and heterochromatin. And there can be histone modifications. And we talked about methylation a bit, and mostly about acetylation. And continuing on talking about eukaryotic gene expression and moving on from the chromatin, we got into the discussion of epigenetics, what that is, and then about DNA methylation at the CBG residues, which is a major factor in epigenetics. And then we talked about RNA polymerase 2 and general transcription factors and how transcription initiates with those. And then about specific regulators and specific transcription factors and the concepts of enhancers and silencers and insulators, and went through some of the basic structures of transcription factors and how they have domains and so forth. And then the final part, I talked about RNA processing in eukaryotes. So that's it. Thank you all very much. And I want to thank my live audience, Sidoni, who's shown here.